Good evening, guys, and today we will start the series on glycolysis. It's a biochemistry playlist, so let's get started. First of all, what is glycolysis? It's the metabolic pathway in which the glucose undergoes a series of steps to form pyruvate, and doing this, we obtain ATP. It's an enzymatic reaction because enzymes are always used, and most of the enzymes are present in the cytosol of the cell. So it's a cytosolic pathway according to the enzymatic location. There are two types of glycolysis. First is aerobic, and the next one is anaerobic. As the name suggests, that aerobic glycolysis occurs in the presence of oxygen, and mitochondria is also used, and in anaerobic glycolysis, there is absence of oxygen in mitochondria. In aerobic glycolysis, the pyruvate is produced and it enters the citric acid cycle. We'll discuss the citric acid cycle in the upcoming videos, but now just remember that in aerobic glycolysis, pyruvate is produced from glucose and it enters the subsequent Krebs cycle. And most of the cells in our body uses aerobic glycolysis for their functioning, and anaerobic glycolysis is used in those cells which lack mitochondria, for example RBCs and the retinal cells, corneal cells. The cells of our retina and cornea do not have mitochondria, so they uses anaerobic glycolysis in which the pyruvate is converted into the lactate. Pyruvate does not go into the Krebs cycle in anaerobic glycolysis. So, first step of glycolysis is glucose acquisition. It means that first step is to obtain glucose from our meal, from the extracellular environment. We obtain a carbohydrate-rich meal, for example, and our meal gets absorbed into the GIT, and from there it is absorbed into the portal vein, which carries the all of the essential nutrients into the liver. It is carried by the portal vein into the liver. It contains all of the carbohydrate monosaccharides, for example glucose, galactose, fructose, sucrose, all of them, and it goes into the liver. And what are the functions of hepatocytes? Hepatocytes, they can store the glucose in the form of glycogen. Okay, they can store the glucose, and when the blood sugar level is high, in case of the rich carbohydrate meal, our blood sugar level spikes, and in this condition, the liver hepatocytes stores the glucose in the form of glycogen. Okay, it's a very important term you need to remember. And glycogen is being produced and the glucose is being stored, so this term is known as the glycogenogenesis. Okay, glycogenogenesis means that formation of glycogen from glucose. And why we need this step? We need this step to prevent hyperglycemia, because we have just consumed a carbohydrate rich meal and we do not want the blood sugar level to rise above the normal certain level. So in this condition, we need to store the glucose as glycogen, this glycogenogenesis. Also, when, for example, in conditions when our blood sugar level suddenly drops, or we have not eaten for our 8 hours, example, our liver hepatocytes are capable to combine the glucose and fructose and form the glucose, and they can release them. Also, they can release the stored glycogen. They can also release the stored glycogen, and the process of breaking down the glycogen is glycogenolysis, and it prevents hypoglycemia. In case of low sugar level, we need to break down the stored glycogen, so it's glycogenolysis. Okay, after that, we need to transport the glucose. Okay, and what is the concept about glucose concentration and transport? Just imagine that it's the bloodstream from which the blood is flowing, and it's the extracellular space, and this inside is the intracellular space. Remember that from the blood capillaries into the extracellular space here, the glucose move freely by the diffusion. The concentration of the glucose inside the bloodstream, inside the blood, and the in interstitial space or extracellular fluid is approximately same. And if there is any change in the concentration, it is balanced by the diffusion of glucose. So just imagine that we have taken a blood sample, blood test for the measurement of glucose. It can be a sugar test. What we are measuring? We are measuring the glucose concentration inside the blood and the extracellular space because these are both the same. But when the cell membrane comes into discussion, the glucose cannot move the freely between the extracellular space and intracellular space. It must require a transport mechanism for its transport between the extracellular space and the intracellular space. And it's a very important to understand that glucose requires special transport mechanism for its transport intracellularly. So coming up, the discussion is about the types of glucose transporters. Glucose transporters, they are named as glut because they are glucose transporters. And let's discuss what is the process. It's the facilitated diffusion. All of the glucose transporters which are glut, mentioned by glut, for example 3, 5, 7, anything, so these undergoes a process of facilitated diffusion. Facilitated diffusion. Diffusion means that it's a passive process. It does not require ATP, and the glucose just move from the high concentration gradient to the low concentration gradient from the interstitial space into the cell. It's a passive process and it's a facilitated diffusion. What's mean facilitation? It means that something is facilitating the process. uh, Seeing this diagram, that it's the extracellular space and it's the intracellular space. We need to move the glucose molecule from this space to this space. So we need a transport mechanism. These are the transport proteins. These are the carrier proteins. These are the glut transporters. And from where they are produced, our cell contain nucleus, and inside the nucleus there is DNA, and DNA contains genes. The genes contain special proteins. They are via the process of central dogma. As you all know that proteins are produced from transcription of the RNA from DNA and from translation from RNA to proteins. And these proteins traverse the membrane of our cells, and these are transmembrane proteins. They facilitate the glucose movement. When we need to transport a glucose molecule, it is attached towards the outer domain of the protein. Then once it's attached, it then flip inwards, and flipping inward releases the glucose molecule inside the cell. So it's a very simple process, but the main thing you need to remember is that the glucose transporter is a facilitated diffusion process between the interstitium and the intracellular space. Coming towards the what are the types of glucose transporters, there are approximately 14 types, but the most important one are being discussed here. And let's get started. First of all is the glucose transporter 1 and 3. I just abbreviate them GLUT 1 and 3. Remember that 1 and 3 are present in most of our cells, most of the tissues, the neurons, central nervous system, RBCs, red blood cells, and placenta. And these 1 and 3 are very, very important because they have high affinity for glucose. It means that, for example, take an example that neurons are very, very sensitive to the glucose concentration. So what kind of transport we need in the neurons? We need 1 and 3. They both can work, and They can easily transport glucose into the neurons, into the RBCs, across the placenta, and they have high affinity for glucose, and they facilitate the basal uptake of glucose. What means that basal uptake of glucose? It means that under the normal low blood glucose conditions or concentrations, when for example we have eaten a meal about 3 hours ago and our blood glucose is normal, just low normal conditions, even in this condition the glut 1 and 3 are still working. About the GLUT2 transporter, it's a two-way transport as the number suggests. The GLUT2, it's a two-way transporter. And what kind of cells in the body think logically would need GLUT2? They should be liver hepatocytes, because the liver hepatocytes need to store the excess amount of glucose as glycogen. So, and in case of hypoglycemia, when we are in low blood sugar level, they releases the excess glucose from their surfaces. So liver hepatocytes should have GLUT2. Enterocytes, our cells of the intestine, small intestine, have GLUT2. Kidney, they also have GLUT2, and the beta cells of pancreas. Why beta cells of pancreas should have GLUT2 transporters? Because as you all know that beta cells of pancreas are responsible for secreting a hormone which is known as insulin, and insulin secretion requires a very sensitive information for glucose. For example, when our blood sugar level increases, the beta cells immediately recognizes and this starts to release insulin. And in this condition, they must require the glucose transporter too, because they are very important in estimating the blood glucose level. Coming towards the next part is the GLUT4, and GLUT4 is very important in skeletal muscles, cardiac muscles, and adipose tissues. Skeletal, cardiac, and adipose tissues, and these are insulin dependent. This is a very important information. You need to remember that GLUT4 are insulin dependent. It means that they require insulin. They require insulin for the glucose uptake, for the glucose transport. So this is an MCQ, just a sort of question that what insulin dependent glucose uptake occurs in all except, what would you say? That skeletal muscles is insulin dependent? Yes, it is. Cardiac muscles are also insulin dependent? Yes, they are. And hepatocytes, what would you think? Yes, hepatocytes are also insulin dependent, but they are indirectly insulin dependent. But here the correct answer is endothelium. Endothelial cells are not insulin dependent for the glucose transport. Why hepatocytes are depending on insulin? Because as you know that when the glucose enters the hepatocytes, it is immediately converted into the glucose 6 phosphate. Glucose 6 phosphate is the storage form of glucose. It means that when the glucose is in this state, it is stored and it is trapped. It cannot move outside the cell, and this process is facilitated by an enzyme known as glucokinase. Okay, and when I say that hepatocytes are indirectly dependent on insulin, it means that glucokinase has its effect because of insulin. Insulin hormone influences the glucokinase to convert glucose to glucose 6 phosphate. So, in this aspect, the glucose transport in liver is also insulin dependent, but it's indirectly. Okay, let's talk about the tissues which are independent of insulin. We have talked about the tissues which are dependent on insulin, the cardiac muscles, the skeletal muscles, and adipose tissues. But what are the cells that are independent of insulin? These are lens, the cells of our lens, the endothelium. kidney, and neurons. These are independent of insulin for their glucose transport. Let's talk about the complications of chronic diabetes. What is this? It means that in case of diabetes, when there is diabetic patient, his insulin level is very low. Okay, the insulin level is very low, and in case of the cells which are independent of insulin, it means that the cells, cardiac muscles, skeletal muscles, hepatocytes, and adipose tissues cannot take the insulin. Okay, because these four cells, cardiac cells, skeletal muscles, and hepatocytes, and adipose tissues, these are dependent on insulin, but these cells, lens, endothelium, kidney, and neurons, are independent. In this condition, there is hyperglycemia in diabetes, so the cells of lens would get affected. How it would get affected? The glucose in our lens is converted into an alcohol, and the alcohol is known as sorbitol, and sorbitol is a polar compound. It attracts the water, and when it attracts the water, it results in diabetic cataracts of the lens. So very important MCQs. It's often asked that how glucose is converted into sorbitol. It's by an enzyme known as aldose reductase in the lens. It converts glucose into sorbitol, and it attracts water. It results in the diabetic cataracts of the lens. The next effect of hyperglycemia is on endothelium. Endothelium results in micro and macrovascular complications of diabetes. As you know that capillaries, our arteries, our arterioles, they are comprising of endothelial cells. So they result in the complications of diabetes. Neurons, neuropathy, and kidney nephropathy. Diabetic nephropathy and diabetic neuropathy are complications of hyperglycemia when the insulin level drops, and these tissues suffer from the hyperglycemia. Okay, we were talking about the glut transporters. Okay, the glut 4 is insulin dependent. What is the function of glut 5? Glut 5 is not a glucose transporter, it's a fructose transporter, and fructose is sweet. And which kind of cells uses fructose? It is the sperms and testes. The cells of intestine, the sperms and testes uses fructose for their metabolism, for their energy. Okay. After that is GLUT7. It's not very important, but just remember that GLUT7 is present in ER, endoplasmic reticulum, and it's involved in gluconeogenesis. You know that gluconeogenesis is a process in which glucose is formed from non-carbohydrate sources, for example, fatty acids and amino acids. And this process occurs in liver and kidneys. In case of gluconeogenesis, we use this GLUT7 transporter. As I have mentioned that why beta cells of pancreas need GLUT2, because they must be very sensitive to the blood glucose level, because they are responsible for the insulin secretion according to the blood glucose level. Okay, what kind of transporters are insulin dependent? These are GLUT4. As you remember that GLUT4 are insulin dependent. So let's make it easy that why insulin dependent? If the blood glucose level rises, pancreas release insulin, and insulin comes towards the adipose tissues, the skeletal muscles, and the cardiac muscles. So these uses the insulin. It triggers the formation of GLUT4. They promote the formation of vesicles. They promote the formation of vesicles that contain GLUT4. For example, these are the vesicles inside of which the GLUT4 is present. But insulin triggers their way towards the cell membrane, and they fuses with the cell membrane and express as GLUT4. This is called as the GLUT4 expression on the surface of the cells. And what is the function of GLUT4? It's the glucose uptake. So indirectly or directly, these GLUT4 are dependent for their expression on the hormone insulin. Okay. There is also questions which are frequently asked, that what is the storage form of glucose or trapped form of glucose? It's glucose 6-phosphate. Whenever the glucose come inside the cell, it is trapped by the enzyme glucokinase and hexokinase, and it's the glucose 6-phosphate. The active form of glucose is UDP glucose. What is UDP? It's uridine diphosphate glucose. This is an active form of glucose. You must remember, physiological form of glucose is beta D-glucopyranose. This is the physiological form of glucose. So storage form of glucose is glucose 6-phosphate, active form is uridine diphosphate glucose, and physiological form is beta D-glucopyranose. Okay, it's a question that neurons, what kind of transporters are present in neurons? Recalling from the text, it's the glucose GLUT 1 and 3. Okay, 1 and 3 are present in the neurons because they require constant supply of energy. Okay, there is another thing I need you to remember, that what is the relation between the sodium and glucose co-transport. This is very important because these are present in the glucose absorption in the intestine and glucose reabsorption in the kidney. What is sodium glucose co-transport mechanism? As you all know that sodium potassium ATPases are present in all the cells of the body. And what is the function of sodium potassium ATPases? They transport 3 sodium out of the cell, and in the same time they transport 2 potassium into the cell. Is it active transport or is it passive transport? We uses ATPase. We are using ATP, so it's an active transport. And whether it's primary or secondary, it's primary active transport, because ATP is being used directly here. Okay, ATP is directly being used here for the transport of sodium out and potassium in against their concentration gradient. Okay, so this is the kidney tubule. This is the cell. It's the proximal convoluted tubule cell, and it's an artery passing by. We need to reabsorb the glucose from the lumen into the cell and from the cell into the bloodstream. How this all process occurs? As you know that through the effect of sodium potassium ATPases, what is the level of sodium inside the cell currently? It's low, because most of the sodium is pulled out of the cell. So inside, the concentration of sodium is very low. So in the luminal membrane means that the membrane between the tubule and the cell, they have special transport mechanisms, and they transport sodium inside the cell. And when the sodium is transported inside the cell, it's a passive process, because the ATP is not used here. Here we are not using ATP directly. We are using the concentration of sodium which is provided by this pump, and we are using that concentration to transport sodium inside the cell. And in transporting the sodium, let's take glucose with it. Okay, and when we take glucose with the sodium, it's also it's coming along with the sodium. It's called as secondary active transport. What is secondary active transport? It means that In primary active transport, we uses ATP directly where the pump is located, okay, and we transport sodium out and to potassium inside the cell. But in secondary active transport, we are just moving the sodium along their concentration gradient, and we are not using ATP here, and we are moving the glucose along with the sodium. It's the secondary active transport which uses the concentration gradient of ions to facilitate the movement of another molecule with them. So from the lumen into the cell, sodium moves passively, okay, here sodium moves passively, and from the lumen glucose moves via the secondary active transport mechanism, okay. It's a very important concept in transporting the sodium and glucose. Okay, there is the last condition. It's a clinical scenario. I need you to guys remember that, just focus on the question. It's in an early morning run, 29-year-old man consumed carbohydrate rich breakfast. It means that we are consuming a lot of glucose. What would you most likely to be activated in the liver? What would be most likely activated in the liver? Is it cytoplasmic Pepsi K? Pepsi K is phosphoenol pyruvate carboxykinase. Is it membrane Glut4? Is it cytoplasmic phosphofructokinase 2? Or is it cytoplasmic glycogen phosphorylase? What is the correct answer? I've mentioned the correct answer, but what is the logic between it? What is the logic behind them? It's that cytoplasmic Pepsi K is involved in gluconeogenesis. Gluconeogenesis means that we are forming the glucose from non-carbohydrate source. After carbohydrate-rich breakfast, do we need gluconeogenesis? No, we do not need gluconeogenesis. Okay, after that is GLUT4. GLUT4 is insulin dependent, okay, and it is not present in the liver. It has influence indirectly. Insulin has influence indirectly, but GLUT4 are not present in the liver, so if it is excluded. Glycogen phosphorylase. Glycogen phosphorylase is used for in the process of glycogenolysis. Glycogenolysis mean that we are breaking down the glycogen. Do we need it after a carbohydrate rich meal? No, we do not need the breakdown of glycogen. Okay, the last option is the phosphofructokinase 2. Phosphofructokinase 2 is a very important enzyme. It is used in the glycolysis, and the function of it is to convert fructose 6 phosphate to fructose 2,6 bisphosphate, and it's an enzyme that promotes glycolysis. Do we need it? Yes, we need to store the glucose. We need to convert the glucose into pyruvate, and we need to generate ATP from it. So yes, cytoplasmic phosphofructokinase 2, it's the correct option after we consume a carbohydrate rich breakfast in the morning or any time in the day. I hope you get the concept about the glycolysis. It's just an introductory lecture, and in the subsequent lectures, we'll discuss in detail about the steps of glycolysis and about the enzymes as well. So that's for today. I will see you in the next video. Goodbye.